a great opportunity to share knowledge and also be exposed to, other parts of the world and to many more people than we might if we were in person. So what I'd like to do in this talk is provide you really a historical perspective and then bring you up to the current state of the field to really illustrate how deep molecular insights can now be translated into novel clinical trials and new therapies. The title of my talk, Remembrance of Things Past, is really taken from the work of French novelist Marcel Proust, who said the only real voyage of discovery consists not in seeking new landscapes, but in having new eyes. And in some respects, this really is pertinent to the talk I'll present, because the work we've been doing in recent years really probes questions that we've had for many, many years that we're now able to address with genetics and molecular science. The general topic then really relates to the hemoglobin disorders, both thalassemia and mostly beta thalassemia and sickle cell disease. These are inherited disorders, obviously distributed worldwide, as shown in this picture below. But what's of particular relevance is, although these are classified in the United States as orphan diseases for the purpose of encouraging development of new treatments, they're really not rare at all. There are more than 300,000 new births per year just of sickle cell disease in the African continent. And there are probably equivalent number of individuals with beta thalassemia worldwide. In the US, there are about 75 to 100,000 individuals with sickle cell disease, and it's a severe burden on the health system, as I'll come to later. So the two disorders, just as background, the beta hemoglobin disorders are beta thalassemia on the left and sickle cell disease on the right, both mutations in the adult beta globin gene, and the chromosome 11, the beta-like cluster is depicted there with an embryonic gene, two fetal genes, and an adult beta globin gene. In the 1980s, actually, my laboratory and with collaborators worked out the mutations in beta thalassemia, which is shown below, distributed throughout the gene. And these are essentially generic mutations that affect production of beta globin or beta globin messenger RNA. On the right is sickle cell disease, which is a single point mutation at codon 6 from glutamic acid to valine, which renders the beta globin and the sickle hemoglobin prone to polymerize and to so-called sickle within the cell. Sickle cell disease is distinguished by being called the first molecular disease. It was identified more than 100 years ago by the gentleman Herrick, shown on the left. But Linus Pauling in 1949 showed that the hemoglobin protein itself was different than protein in normal individuals, and he dubbed this a molecular disease. And 7 years later, Vernon Ingram identified the specific point mutation. We've known for many, many years there are genetic modifiers for both sickle cell disease and for beta thalassemia. And one of the important modifiers is fetal hemoglobin, which is the alpha chain with the gamma chain, the fetal chain. And it was described actually in 1948 by a pediatrician, Watson, that infants who were destined to have sickle cell disease were not ill in the first several months of their life. And she proposed that this was due to the presence of fetal hemoglobin at high levels, which would prevent symptoms and also in reliance on sickle hemoglobin. And in fact, she was correct. Natural history studies, such as the one on the bottom, conducted more than, almost 30 years ago, showed that individuals with a higher level of fetal hemoglobin, on average, if they had sickle cell disease, had a longer survival. Now, the fundamental problem in sickle cell disease is that the hemoglobin is obviously abnormal. It's a Mendelian single gene disorder, but it has a amazingly variable clinical phenotype due to genetic modifiers such as alpha thalassemia and also the fetal hemoglobin level. And if one wants to address the primary problem in sickle cell disease, or the primary problem, if you will, in thalassemia, one has to address the nature of the hemoglobin. And in terms of sickle cell disease, one could directly interfere with polymerization if you had a chemical that could do that. You could shift the oxygen dissociation curve, and that's the basis of a drug, Valoxator, which was just recently approved in the United States from Global Blood Therapeutics in California. Or one could change the hemoglobin. And from a genetic standpoint, one could repair the mutation in the hemoglobin, provide a new hemoglobin that would not sickle, or provide fetal hemoglobin. There are a number of possibilities. In the case of beta thalassemia, where there's less beta chain and less adult hemoglobin, you could provide a hemoglobin, either adult hemoglobin or fetal hemoglobin. This is the really central focus of much of our work over the last several years, and it's been to understand how the developmental switch from fetal to adult hemoglobin occurs. This is important because the major mutations in thalassemia and sickle cell disease occur in the adult beta globin gene, and therefore the diseases are those of adult stage red cells. If one could turn on and reverse the expression of the globin such that one made fetal hemoglobin in the adult stage rather than adult hemoglobin, one would be able to correct these disorders entirely. And this has been the goal of the field for many, many years. The level of fetal hemoglobin in adults is genetically determined. In normal individuals, it's around 1% of the total hemoglobin is fetal hemoglobin, and that's largely restricted to a small population of cells called F cells, which contain that fetal hemoglobin. The goal of much of work over many years in the field has been to increase the level of fetal hemoglobin from this 1% to something on the order of perhaps 20%, so-called therapeutic level, which in sickle cell disease would prevent sickling of the hemoglobin. In the case of thalassemia, would replace the missing adult hemoglobin. The major obstacle in achieving this has been a lack of understanding of how the switch from fetal hemoglobin to adult hemoglobin is actually programmed. Many years ago, actually in 1982, Timothy Lay and Arthur Nienhuis attempted to turn on fetal hemoglobin with the demethylating agent aza-C, based on the notion that the level of DNA methylation would control the level of fetal hemoglobin. They achieved a small increase in hemoglobin F, but because of concerns of toxicity of long-term administration of the agent, the clinical application did not proceed any further. Around the same time, Aura Platt at Children's Hospital, in collaboration with myself and David Nathan, introduced hydroxyurea as a potential agent. It induced fetal hemoglobin to some degree, and the rationale was to somehow perturb cell cycling. Hydroxyurea does increase hemoglobin F variably in sickle cell patients, and it's now the standard of care throughout the world. But there's no good explanation of how this occurs, and many patients do not respond well to hydroxyurea. So it's really not a totally successful approach to therapy. So the questions I want to address in the next few minutes then is really, how is this hemoglobin switch controlled? What are the nuts and bolts of the part list? What are the regulators? What's the mechanism of the switch, and looking at detailed mechanism? How can our improved understanding of the switch be leveraged to develop new strategies for therapy? And then what challenges remain? So in terms of the nuts and bolts of the parts list, the major breakthrough came a little more than 10 years ago with the application of genome-wide association studies to analyzing the level of fetal hemoglobin. These studies are performed in populations with SNPs across the genome. There were two major studies performed, one by Swilatheen in the UK and another one by Antonio Cao and colleagues in Sardinia. They both arrived at the same findings, and that is that there are three regions of the genome in which loci can be associated with change or elevation of fetal hemoglobin in the normal population. It's not a diseased population. These were on chromosome 11 with a beta cluster, on chromosome 6 with a MIB oncogene lies. But the new factor, and the one of central importance here, was on the short arm of chromosome 2, BCL11A. These are called Manhattan plots, and in this case, I put the Empire State Building, tall building in New York City, right over BCL11A. If one sums the genetic contribution of these three loci, one can account for more than 50% of the genetic variation in hemoglobin levels, hemoglobin F levels overall, which is really an enormous contribution for these kinds of genome-wide associations. So we began to focus on BCL11A as a candidate regulator of hemoglobin F. And the challenge in GWAS studies then is to go from the entire genome of 3 billion base pairs down to a relevant target gene or sequences that can account for the trait or the disease, and this is the major goal. We've done that over a series of experiments over several years, in which we first focused on validating the gene BCL11A as a regulator of fetal hemoglobin. We then sought to understand how it acts and identified an enhancer within the gene that's critical for its regulation, and then focused on how to apply this information using gene editing and other approaches, as I'll come to. The initial evidence that BCL11A was in fact a potential regulator of fetal hemoglobin came from this experiment by Vijay Sankaran when he was a student in the lab, in which we used the CD34 differentiation system and inhibited BCL11A with an shRNA. We recognize that BCL11A is adult specific, and it's only turned on in the definitive hematopoietic lineages in mouse and human. Mouse only has one switch from embryonic to adult. Human has switched from embryonic to fetal to adult. And one can see on the Western blot shown on the bottom left that BCL11A is not expressed in the primitive lineage that's derived from the yolk sac. In the CD34 system, as shown in the purple, shRNAs to BCL11A increase the level of gamma chain expression, that is the beta-like chain in fetal hemoglobin, and this at least provided some suggestion that BCL11A is in fact a repressor or regulator of gamma chain expression. And we suggested at that time, this was 2008, that in fact BCL11A might be a therapeutic target for reactivation of fetal hemoglobin. That was sort of a wild speculation at that time. So how important is BCL11A as a single factor? If many factors regulate fetal hemoglobin expression, manipulating a single factor might not in fact have much relevance to any therapeutic application. We turn to the engineered mouse models of sickle cell disease, one of which is shown here, the Berkeley mouse, which is a mouse in which the mouse globin genes are ablated by homologous recombination, and human genes have been introduced as transgenes such that they're expressed in a roughly normal pattern, and in this case, sickle beta globin gene. We then performed an experiment in which we used conditional BCL11A knockout mice and used erythropoietin receptor CRE to delete BCL11A only in the erythroid lineage. And in mating with the sickle mice, we asked whether this could rescue the phenotype, that is, correct the mice. And as shown here, the result was actually quite remarkable and perhaps better than we ever expected. A knockout of BCL11A indeed rescues these mice nearly completely. As shown on the top of the blood smears, you can see the sickle smear in the middle with the sickle forms. And in the mice lacking BCL11A and the erythroid cells, the smear is normal. The hematologic parameters are essentially normal. And by FACS analysis, as shown on the right in the middle, more than 85% of the cells contain substantial amounts of fetal hemoglobin. And as shown on the bottom left, this amounts to roughly 25 or close to 30% of the total hemoglobin in these mice. And that's why, in fact, they're rescued, because there's enough fetal hemoglobin to block the sickling process and also replace a portion of the beta chain expression that's devoted to the sickle gene. So removal of BCL11A in the erythroid lineage is sufficient to abolish sickle cell disease through reactivation of fetal hemoglobin. And this experiment performed by Jan Zhu in 2011 really is the proof of principle that this is a genetic target for therapy. The field has benefited most recently from the use of cell lines, which were created to essentially immortalize CD34 cells. These are CD34 cells that have been transformed with an oncogene, and they come in two varieties, HUDEP2, which is an adult type line expressing human hemoglobin A, and HUDEP1, a fetal line which expresses hemoglobin F. And although these are transformed cells, they phenocopy normal CD34 erythroid-derived cells quite closely. And particularly, they can be used as targets for gene editing and gene manipulation. One can clone these lines and really perform genetic experiments to test mechanisms of action. BCL11A is not the only repressor. There's a second repressor, which was identified by Taka Mieda when he was in Boston, and that's known as LRF or ZBT7A or Pokemon, and it's roughly equivalent in power as a repressor as BCL11A. If one knocks out either one of these proteins in HUDEP2 cells, one gets approximately 50% fetal hemoglobin. And if you knock them both out, one gets essentially 100%. And we believe these are the two major repressors of fetal hemoglobin. These two repressors each interact physically with the NERD chromatin remodeling complex, which is a multi-protein complex shown in the middle. They interact with these proteins using different domains, but physically interact. The two major repressors then are BCL11A and LRF. Both interact with this complex. And there are selective requirements, however, for alternative subunits within the NERD complex, work of my colleague Dan Bauer in Boston. And the knockout of both repressors leads to essentially 100% fetal hemoglobin. So the real crux then is, how does one engineer this switch from fetal hemoglobin to adult hemoglobin? And the LCR contacts the downstream genes. The LCR is a super enhancer, the first described super enhancer, in fact, and it contacts the genes directly through chromosomal looping in the fetal stage, touching the gamma genes, activating them for expression. The beta gene is excluded for the LCR and is silent. And then BCL11A and LRF repress the gamma genes, preventing LCR interaction and function, and the LCR is then free to activate beta globin gene transcription. But how does this work in detail? What's the mechanism of the switch? In order to get at that, we needed to know more about the BCL11A protein. The protein is about 100 kilodaltons depicted at the top. And in the C terminus is a cluster of 3 zinc fingers called zinc fingers 4, 5, and 6. And with Martha Bullock at Harvard Medical School, we determined that this cluster interacts with and recognizes the DNA sequence TGA-CCA, which is a unique binding motif, which was not in the transcription factor database. And we've produced crystals and determined the crystal structure of this region on DNA as shown below. And the critical point is that figures 5 and 6 confer most of, figures 4 and 5, sorry, confer most of the binding potency to this TGA-CCA sequence. We also wanted to know where BCL11A actually binds within the genome, and we'd use conventional ChIP-Seq approaches using cross-linking and antibodies to BCL11A, but we found over several years and attempts by several individuals in the laboratory that ChIP-Seq does not work well for BCL11A. It gives you spurious results. You get peaks, but they don't encompass this TGA-CCA binding site. We then turned to a newer procedure devised by Henikoff in Seattle called CUT&RUN, which is essentially a native kind of ChIP assay in which an antibody is recruited to a protein A micrococcal nuclease fusion. And this produces much smaller fragments and avoids any cross-linking in the procedure. The benefits are that it is less manipulation, there's no cross-linking, no sonication, the resolution is higher, and this is a procedure that is gaining acceptance as an alternative to ChIP-Seq. We found using this that we could perform cut-and-run on CD34 cells and HUDEP cells and identify de novo the TGA-CCA motif and footprint that region as illustrated here. And this is the work of Nan Liu in the laboratory, who was actually trained initially in Beijing, and Jian Zhu, who was our computational biologist. As a point that might be useful for many investigators, Chan developed a pipeline for analysis of CUT&RUN called CUT&RUN tools, and this software package allows motif discovery, footprint analysis, and single locus footprinting, which essentially gives you a nucleotide resolution of the binding of factors to chromatin. We use CUT&RUN for the first time then to identify that BCL11A bound directly within the gamma promoters, and this is a shot of the whole beta globin gene cluster, and you can see with the pink highlighting where the gamma genes are, and BCL11A binds directly. And this was really quite informative and important because this region of the promoter had previously been identified as a site of point mutations in the syndrome called hereditary persistence of fetal hemoglobin, or HPFH, which was identified in 1985 in work of Francis Collins at the time and George Stamanianopoulos. And within the TGA-CCA sequence, there are naturally occurring mutations which lead to increased fetal hemoglobin, 10 to 30 times normal, such that in the adult there's significant level of fetal hemoglobin. At the time in 1985, these mutations were thought to affect the CAT box, CCAAT, which is abutting the BCL11A site. In addition, as boxed on the right, there's a second TGA-CCA sequence and a second CAT box located more proximal in the gamma gene promoter. And I'll come to that in a second. That's not the site of any known mutations. We could explain the HPFH mutations because the zinc finger 5 drives recognition of the core of TGA-CCA, and the mutation occurring in HPFH interrupts that interaction. On a very high resolution view with CUT&RUN of the gamma promoter, as shown here, we saw that the peak of BCL11A, the peak associated with BCL11A was actually shifted to the distal duplicate of the TGA-CCA, as shown here, which suggested that BCL11A binds the distal rather than the proximal site. And this summarizes this conclusion, which is supported by other data that some of which I'll mention, but don't have time to fully go into, that there's a duplication of TGA-CCA in the promoter. BCL11A in adult cells represses through the distal site and recruits the NERD complex, which then enzymatically really facilitates repression and closure of the locus. So what drives gamma gene expression in the fetal stage? Well, we surmised that it was most likely NFY, which is a major CAT box binding protein, and we could see by CUT&RUN that it binds in fetal expressing cells, but not in adult cells, which are not expressing fetal hemoglobin. We could demonstrate a footprint at the where NFY binds. And importantly, we could even do single locus footprinting, which at the bottom revealed that the true NFY footprint is actually over the proximal CAT box and not over the distal CAT box. We also used more recently dense CRISPR-Cas9 tiling across the whole beta globin cluster with Cas9, dead Cas9 with an activator or repressor module attached. I don't have time to review all the data, but what we could show was that recruitment of dead Cas9 to the BCL11A binding site, in fact, reduces gamma globin expression rather than activating gamma globin expression, as one might think. And if one looks out at -200, where LRF binds, if one places DCAS9 at that position, one gets an increase in fetal hemoglobin, which is consistent with displacing a repressor. But in the case of the distal TGA-CCA binding site, displacement with dCAS9 led to a lower level of fetal hemoglobin, which at first was a paradox. But we've resolved that by detailed study using base editing of both the proximal cat box, as shown here, or the distal TGA-CC motif. In this case, if we base edit the proximal site of the cat box, we reduce fetal hemoglobin expression, which is consistent with NFY binding and being the activator. And if we do the converse and base edit the distal motif, one gets more fetal hemoglobin, and that's consistent with that site being the site of repression by BCL11A. And so what we've concluded is that placement of dCAS9 within the proximal promoter of gamma globin, particularly right over the BCL11A binding site, displaces not only BCL11A, the repressor, but NFY, the activator. So that's why one gets a paradoxical result. And so in the fetal stage, we believe that gamma globin expression is driven by NFY and whatever co-activators bind it. However, in the adult stage, the repressor BCL11A comes on, recruits NERD, and represses the locus. And what's remarkable about this is this kind of competition between a repressor and activator occurs within a 21 base pair region of the promoter. And we believe this is steric hindrance mediated through BCL11A. So BCL11A, we believe, has two roles. One is to directly repress at the promoter by physical obstruction or hindrance, but also by recruiting the NERD complex, it facilitates repression and closure of the locus. This is then a sort of high resolution summary of the mechanism of the switch, with BCL11A binding at a repressor site, the TGA-CCA sequence, the LRF repressor binding further out. Both of these binding sites are the sites of naturally occurring mutations in HPFH. Both of these recruit a specific NERD complex, and BCL11A displaces the activator protein NFY during the switch process. So the switch is really locally controlled despite long-range chromosomal interactions. Now, I mentioned before the challenge presented in GWAS is how one goes through 3 billion bases to only a few bases to a relevant target. Dan Bauer, when he was a fellow, identified within the BCL11A gene and intron 2 DNase hypersensitive sites that really are overlaid by HPFH-associated SNPs that have been identified in GWAS studies. And if we blew up this region, it's really more than a 10 kilobase erythroid-specific regulatory element, an enhancer, with these 3 hypersensitive sites. We predicted at that time that this enhancer would be a useful target for gene editing because it's erythroid-specific. And that's particularly important because BCL11A is expressed in other tissues, and we'd want to restrict effects as best we could to erythroid cells. Deletion of the enhancer, in fact, shows that it's absolutely erythroid-specific. And that's illustrated in this experiment, which I'll skip over for lack of time. We went on to analyze the enhancer in greater detail using CRISPR-Cas9 dense mutagenesis with pooled library as illustrated here. And this led to the remarkable finding that within this 10 kilobase or slightly more than 10 kilobase enhancer, there's an Achilles heel, a site right at the GATA1 site within the hypersensitive site known as +58, which when disrupted leads to a marked impairment of enhancer activity, probably 2/3 to 3/4 of the entire activity of this 10 kilobase enhancer is mediated through that single GATA site. And that site then has become the focus of gene editing efforts for therapy in the beta globin disorders, as I'll summarize shortly. BCL11A is really quite unique in genetics because common genetic variation, GWAS, and rare genetic variation through HPFH converge on the same protein, one being the enhancer, regulation of the repressor, and the other, the specific cis regulatory binding site of the repressor protein itself. So the genetics here is extremely powerful. And this summarizes really the genetics of BCL11A and relevance to therapy. In so-called normal individuals with a common low hemoglobin F haplotype, BCL11A represses fetal hemoglobin, and one has essentially hemoglobin A in the adult cells. In those individuals who have a common genetic variation, which leads to a slightly higher fetal hemoglobin, one makes a slightly less BCL11A because of variation within the enhancer. One has a small increase in fetal hemoglobin. So essentially, the cells still largely have hemoglobin A. If you delete the BCL11A enhancer entirely, one has less than 1% BCL11A expression, and one has marked induction of fetal hemoglobin and a reduction in beta globin production. And if one targets the Achilles heel within the enhancer, one gets a markedly reduced level of BCL11A. One still gets marked induction of fetal hemoglobin, and this would be of benefit to patients with beta thalassemia or those with sickle cell disease. So how can we apply this information? Well, we can leverage these findings for genetic therapy to destroy or reduce the level of BCL11A in erythroid cells, and this must be accomplished in hematopoietic stem cells as somatic gene therapy. And the possible approaches then are to knock out the BCL11A gene, knock it down, edit the enhancer, or mutate a binding site for BCL11A, for example, in the gamma globin promoter. The knockout of BCL11A entirely is toxic for hematopoietic stem cells and B cells, so it's not the optimal strategy. shRNA, particularly in erythroid-specific manner, could be very useful, and that's an ongoing trial at Boston Children's Hospital that I'll summarize. One can edit the erythroid enhancer BCL11A with either zinc fingers or CRISPR-Cas9, and those are trials that are ongoing. And one can mutate a binding site in the gamma globin promoter. These are preclinical studies in a number of laboratories. So BCL11A is essentially a regulator of fetal hemoglobin, but what level of inhibition do you actually need to be successful for therapy? What's particularly instructive here are rare individuals with an intellectual disability syndrome where they're haploinsufficient for BCL11A. Either they delete the gene or have mutations in the gene. These unfortunate individuals have severe problems in speech and development, but they have a level of fetal hemoglobin on average that's around 13 to even 20% or more, which is in the range of the classical hereditary persistence of fetal hemoglobin, sufficient to ameliorate significantly sickle cell disease or thalassemia. This is a 50% reduction. Our estimate that a somewhat greater reduction, around 60%, is sufficient to be therapeutic in sickle cell disease and in thalassemia. One can then conceive of using genetic therapy, using modification either by lentiviral gene therapy or gene editing of hematopoietic stem cells, and reintroducing these modified cells into individuals who have been conditioned, their host marrow destroyed, and then one can reconstitute hematopoiesis with these modified cells. There are many critical parameters to consider in these kinds of experiments. I don't have time to review these, but the most important ones have to do with the efficiency of the modification and the details really of bone marrow transplantation. The trial at Children's Hospital, which has been ongoing for the last 2 years, led by Dr. David Williams, as shown on the bottom left, uses an shRNA to B11A embedded within a lentivirus that is expressed specifically in erythroid cells due to the presence of globin regulatory elements derived from the LCR. And in this trial, the patient's cells are harvested, treated, frozen. The patient undergoes conditioning to destroy the host marrow, and then they receive an infusion of the modified cells. And the first patient treated was an adult sickle cell patient shown on the bottom right. And he is now more than 2 years post-therapy. This patient is shown here, this first patient, time course. In the bone marrow, his level of B11A protein is markedly reduced by the shRNA. Over the course of following the gene therapy, his level of F cells, the cells in the blood containing hemoglobin F, rose to approximately 70 to 75%, where they've been plateaued for the last 2 years. His total level of fetal hemoglobin is about 25%. His blood smear has been entirely corrected. And he's been asymptomatic, has had no symptoms of sickle cell disease, and he's essentially cured. We believe he's cured. His F cells, those 75% cells that are shown in red, contain about 40 to 50% of fetal hemoglobin per F cell, which is more than enough to prevent the sickling process. And what's remarkable about this trial, as shown here, is that in 5 subsequent patients that have been treated, the results are virtually identical. All patients have approximately 70 to 75% F cells at steady state. They have a essentially normal hemoglobin level, and all patients have been asymptomatic and free of disease. This clinical trial is in press for publication. At the same time, CRISPR Therapeutics and Vertex, 2 commercial entities, collaborated on a clinical trial using CRISPR-Cas9 editing of the erythroid enhancer at the Achilles heel GWAS site that I described previously. And they've treated both patients with beta thalassemia and sickle cell disease. And they've recently released findings on these patients in an abstract for the upcoming American Society of Hematology meeting. And this is representative of the sickle cell patient who was treated, who at this stage was 9 months post-therapy and had more than 46% fetal hemoglobin and essentially 100% F cells as shown on the top. And this is representative of a patient with transfusion-dependent beta thalassemia who was able to achieve a steady state high level of hemoglobin, which is essentially all fetal hemoglobin. And by report, the patients they've treated thus far, approximately 5 beta thalassemia patients and 2 sickle cell treatment patients have had excellent results and are transfusion dependent and free of symptoms, equivalent to the shRNA trial. So this is the state of clinical trials of genetic therapies. There are ongoing trials from Bluebird Bio on globin addition therapy, which is to use a lentivirus to provide a non-sickling beta globin, either beta thalassemia or sickle cell disease. For beta thalassemia, this approach is approved already in Europe. There are other globin addition trials ongoing in the US, but hemoglobin F reactivation, the first trials are shown below, which are the Children's Hospital trial, which was done in conjunction with Bluebird using an shRNA, and then the CRISPR editing trial of the enhancer shown below in both sickle and beta thalassemia. These are not the only targets for therapy. One can also imagine targeting the BCL11A binding site in the gamma globin promoters, and that's being considered and under investigation by a number of entities. One can also consider using base editing to convert a binding site to an HbFH mutation. And finally, one can consider using homologous repair to correct the beta globin gene, particularly in sickle cell. So we believe now that the genetic therapy has been validated. One can achieve this and it's successful. And this is really the first indication that therapeutic reactivation of hemoglobin F can be achieved successfully. There are a number of things that one needs to do to improve this therapy. One would be to have non-ablative preconditioning to allow for greater sort of expansion of the number of patients who could be treated. And finally, if one could use in vivo gene therapy, one might be able to get to many, many more patients than one can access using really resource-intense bone marrow transplant. So what remains? I think the real challenge is that there are many, many patients, and one can never do gene therapy or editing, at least in the current, by current methods, to affect the burden of disease in a population. And one needs small molecules that might be able to do this. And this is really emphasizes the vision of David Weatherall that one needs to treat these diseases as global health problems and not just as rare disorders. So a vision going forward, I think, is really summarized at the bottom is to see whether we can achieve pharmacologically what we can now do through genetics and perhaps really address the undruggable targets with small molecules. This is a great challenge, but one that is worth undertaking. So in summary, the take-home messages, I think, is the mechanism of this fetal to adult switch is now coming into focus, and BCL11A is a central component. BCL11A is now a thoroughly validated target for therapy of hemoglobin disorders. The clinical trials that are both gene editing and gene therapy have validated all the preclinical science, and we think will lead to approved therapies for these disorders and be curative as well. And I think a challenge for the future is developing on-target small molecule therapy that can really address the global impact of these disorders, a difficult but important problem.